how the drug molecule is there. Then, so the physicochemical properties of that drug molecule are going to either increase the biological action or they may going to decrease the biological action. But before we actually discuss about what is biological action, we have to study the terminologies, that is drug metabolism, how it is metabolized in our body. Now the drug metabolism is also known as biotransformation. Now the meaning of the word transformation means change, and the change done by a biological enzymes which are there in our body. Now, what those enzymes are going to do to that drug molecule, so it is get transformed, it will get metabolized in our body, that we are going to discuss in this particular lecture. Now, what happens when we are going to take a drug molecule? First of all, the drug will be absorbed. First of all, it is digested. It is available unionized form in our stomach, or it is in the intestine, and that unionized form of drug molecule will be available in the bloodstream. Now, the moment the drug molecule is available in the bloodstream, it is going to reach to the site of action. Now, so we are going to discuss the drug metabolism, means what our body is going to do with that drug molecule and what are the effects of that drug molecule with our metabolic systems. Now, already we have discussed. So what happens when the drug is absorbed through our absorption membranes kiwa cell membrane? Cell membrane madun those drug molecule will be available or will go into the bloodstream through circulation. Now blood is continuously moving throughout our body, so it is going to be absorbed from the cell wall to the bloodstream. Now, with the help of this bloodstream, the drug is going to reach to the site of action. Suppose I have taken a tablet of paracetamol for the headache. So what happened? First of all, I am going to take that tablet. It is dissolved in our stomach through stomach cell linings. It is going into the bloodstream through the cell wall of the stomach. Now, after it is going to reach to the site of action, that is head. Now, what happened after that? It is changed so that it can be excreted. When it is going to reach to the site of action, it will going to give. It is going to reach to the site of action. It is going to give its pharmacological action so that the headache is treated, and after that, the drug molecule is changed. See the terminology, it is changed so that after giving its pharmacological action, it can be excreted throughout our body. And after excretion, we know what is excretion. After excretion, it is leaves, it is removed out of our body. So what is drug metabolism? It is a biochemical. What type of modification? It is a biochemical modification of a pharmaceutical substance, and which is done by the specialized enzymes. Those are present in our body. So what are those enzymes, or what actually they are going to do in our body? The drug metabolism or biotransformation means it is a chemical alteration of the drug in the body, and it is needed to render. What type of change the metabolic enzyme is going to do in our body? They may change the lipid soluble compounds into lipid insoluble compounds so that they are not reabsorbed in the renal tubule. See, कसल्या types of modifications करता है biotransformation? After giving its pharmacological action, त्यांनी त्याची pharmacological action दिल्यानंतर the remaining part is not used, or the remaining part is not useful for our body. So that remaining part is changed कशामधून कशामध्ये? लिपिड सोल्युबल कंपाउंड्स किंवा लिपिड सोल्युबल कंपाउंड असेल, ऑर्गॅनिक कंपाउंड असेल, त्या ऑर्गॅनिक कंपाऊंडचं कन्व्हर्जन अशा कंपाऊंड मध्ये केलं जाते की तो लिपिड इनसोल्युबल, म्हणजेच सो दॅट दे आर नॉट रिअब्सॉर्ब, तो पुन्हा एकदा रिनल ट्युब्युल मधून किंवा किडनी मध्ये ऍब्सॉर्ब होऊ नये अशा पद्धतीने त्याचं मॉडिफिकेशन केलं जाते and they are excreted out throughout our body. So this type of modification is known as biotransformation. Now what happened, most hydrophilic drugs कसल्या टाइपचे ड्रग मॉलिक्युल्स, they are more amount of excreted from our body, that is streptomycin, neostigmine. So those type of drugs are little biotransform, त्यांचं थोड्या प्रमाणातच मेटाबॉलिझम केलं जातं and they are largely excreted out as it is, that is unchanged. म्हणजेच our body are not going to change a total change or total biotransformation of कसले drug molecules, streptomycin, neostigmine. Generally those drug molecules किंवा या प्रकारचे drug molecules they are excreted as it is, excreted unchanged from our body. So the primary site of action for drug metabolism is generally हे बायोट्रान्सफॉर्मेशन किंवा ड्रग मेटाबॉलिझम कुठल्या ठिकाणी केलं जातंय, in the large amount it is done in the liver, आणि लिव्हरच्या बरोबरच other metabolizing sites are kidney, intestine, lung and the plasma. So these are the organs which are actually involved in the drug metabolism किंवा biotransformation. आता what type of action the enzymes are going to do on the drug molecule in the biotransformation process. बायोट्रान्सफॉर्मेशनच्या प्रोसेस मध्ये कुठला एन्झाइम कुठल्या टाइपची ऍक्टिव्हिटी शो करतो, that we are going to discuss. For example, there are 4 types of enzymes, A, B, C, D. This is an example. These are not actually enzymes. So the action shown by the enzyme A is it will convert the drug molecule into a metabolite A. Now, with the help of enzyme A, the drug molecule is converted into metabolite A, but it is inactive. See, it is metabolized. It is biotransformed with the help of enzyme A, but after enzyme A reaction, drug is not going to show the activity. It is inactive as such. Now, what happened when a second enzyme, that is B enzyme, is going to metabolize the drug molecule? It will convert the drug into metabolite B. Metabolite म्हणजे converted product. And after the action of this metabolite enzyme B, drug is converted into metabolite B, which is active in nature. Now the next enzyme, that is enzyme C, which will convert the drug molecule into metabolite C, but it will convert the intoxic drug molecule into toxic drug molecule. And finally, there are enzyme types which will convert or which are metabolize that drug molecule, but it will convert them into an undetectable. We don't know what type of conversions they are going to do with that drug molecule. So these type of activities are shown by a different type of enzymes in order to show their metabolizing actions. Now, all these metabolizing actions of biotransforming enzymes are divided into 2 types. सगळ्या biotransformationच्या reactions, those are there. They are divided into 2 types, phase 1 reaction and phase 2 reaction. See, कसल्या टाइपच्या activities the enzyme molecule is going to show, we have seen. It will convert into active metabolite, it will convert into inactive metabolite, it will convert into toxic metabolite, and we don't know what type of conversion, that is undetectable. But those all biotransformation processes are divided into 2 phases, phase 1 reaction and phase 2 reaction. Now, phase 1 reaction is also known as biotransformation. Actual transformation किंवा actual enzymatic reactions या process मध्ये होतात. That's why phase 1 reactions are known as biotransformation reaction. Now, what type of reactions are actually involved in the phase 1 reaction? It will include oxidation, hydroxylation, reduction, hydrolysis. Now, these enzymatic reactions of new functional group are introduced into the substrate molecule. Phase 1 reaction मध्ये through these processes, that is oxidation, hydroxylation, reduction, hydrolysis, a new functional group is introduced. वेगळ्या प्रकारचा functional group is introduced केला जातो in the drug molecule, and existing fractional group is modified. Either they are going to change the functional group, or they may modify the functional group, or the functional group or the acceptor site for the phase 2 transfer reaction is exposed. अशा पद्धतीने त्या ड्रग मॉलिक्यूलचं कन्वर्जन केलं जातं की जेणेकरून फेज 1 रिएक्शन मधला बायोट्रान्सफॉर्म्ड प्रोडक्ट हा इझिली अवेलेबल होईल फेज 2 रिएक्शन साठी. So this is stage. In case of stage number 1, that is phase 1, the drug molecule is either substituted or changed the functional group, or modify the functional group, or the functional group or acceptor size for phase 2 transfer reaction is exposed for making the xenobiotics more polar, therefore readily excreted. Now the meaning of the word xenobiotic means type of reaction those are happening on the drug molecule kiva presence of enzymes which are going to affect the drug molecule. This is known as xenobiotics. Now what happened in case of phase 2? Phase 2 is nothing but reactions are enzymatic synthesis whereby a functional group such as specific functional group such as alcohol, phenol, amine is masked by the addition of new functional group. See, phase 1 मध्ये काय केलं जात आहे? फंक्शनल ग्रुप एकतर चेंज केला जात आहे, नवीन फंक्शनल ग्रुप इंट्रोड्यूस केले जात आहेत, एक्झिस्टिंग फंक्शनल ग्रुप मॉडिफाय केले जात आहेत, जेणेकरून ते फेज 2 साठी इझिली एक्सेप्टेबल होतील. And what happening in the case of phase 2? Phase 2 मध्ये enzymatic synthesis reactions whereby a functional group, specific functional group like alcohol, phenol, amine are masked by the addition of new groups. So they are going to mask these groups because they are polar in nature, and those polar group we are going to mask by either acetyl group, sulfate group, glucuronic acid, and certain amino acid, which are going to increase the polarity of the drug or xenobiotics. Now the most substances undergo generally both type of transformations, that is phase 1 transformation as well as phase 2. Earlierly they are going to for the phase 1 reactions and later on for the phase 2 reactions. Now phase 1 versus phase 2. What type of actual conversions are going to take place in phase 1 and phase 2? Phase 1 involves generation or exposure of functional group, and in this case the metabolite, whether it is from A, B, C, D, may be active or inactive after phase 1. But after phase 2, the metabolite is, phase 2 is also known as synthetic or conjugated reaction. After this, an endogenous radical conjugated of the drug and metabolites from after the reaction of phase 2 mechanisms, they are generally inactive in nature. See, in the case of phase 1, it may be active or inactive, but after phase 2 reactions, it is generally inactive in nature. Now, what type of conversions are going to take place in phase 2? Generally, glucuronide conjugation. Example is glucuronide conjugate of morphine and sulfate conjugate of minoxidil are active. So this is an exceptional case of phase 2 mechanism. Generally phase 2 mechanism नंतर drug molecule कशामध्ये convert केला जातो? Inactive metabolite मध्ये convert केला जातो. But with the case of glucuronide conjugation, it is going to convert it into an active metabolite. Now, a special type of enzyme which is going to play his role in the phase 1 reaction is cytochrome P450 enzyme. Very important enzyme which is going to have his role in the phase 1 reaction. Now, already we have discussed what is going to happening in the case of phase 1 reaction, especially oxidative bio-bond transformation process are most common and important in this type of drug metabolism. That's why we are going to discuss this enzyme. What is this enzyme? This enzyme carrying out the transitions are referred as oxidases and monooxidases. We have already discussed what type of conversion it is going to give, oxidative type of conversion. We have discussed this enzyme in the biochemistry for the electron transport chain. At that time we have discussed this enzyme, and this enzyme is going to give us the oxidative type of mechanism, that is mixed function oxidases or monooxidases. Now, what type of conversion this enzyme is going to do? Suppose this is our drug molecule, then an NADPH molecule which is actually involved in the electron transport chain, it will convert the NADPH molecule into NADP. So the general stereochemistry of oxidation process of drug molecule. So this is happening in the case of electron transport chain, that is release of energy from the So there is large amount of reactions are happening in these processes. There is large family which are going to carry out some basic cytochrome 450 enzymatic reactions on the drug molecule. So this is a family of enzyme named as cytochrome 450. This one belongs to its family. A is its subfamily, and this number in the name of cytochrome 450 is going to give us or going to provide us the information about of each subfamily. So there are subfamily enzymes available in the same family, that is cytochrome 450, cytochrome A12, cytochrome 269 and cytochrome 2D6. So these are the enzymes which are going to give or which are providing these type of reactions. But we are going to give, or the phase 1 reaction is going to utilize only this enzyme, that is cytochrome 450 enzyme. Now, why only cytochrome 450 is used for the phase 1 type of reactions? You are going to find out the answer of this question, that why phase 1 reaction is going to involve the use of cytochrome 450 enzymes. Now we are going to further proceed for the discussion of type of reaction which are involved in the phase 1 reaction. Generally oxidative type of reactions we have already discussed. Now what type of oxidation? Number 1, oxidation of aromatic moieties. See, so many drug molecules which are available for the treatment of n number of diseases, they are going to possess the aromatic ring. आणि त्याच aromatic moieties oxidation, that is phase 1 metabolism करण्यासाठी, or those type of drug molecules are metabolized through which type of process? Phase 1 processes. Then oxidation of olefins, lipid molecules which are used for the treatment. Then oxidation of benzylic, allylic carbon atoms and carbon atoms alpha to the carbonyl and imines. Then oxidation at aliphatic and alicyclic carbons. Oxidations which are going to involve the heterocyclic systems. See, vitamins and all other drugs are used for the treatment which are going to have the aliphatic and alicyclic carbon atoms. They are going to involve the heteroatoms, that is pyridine, pyrimidine, So those are all metabolized by phase 1 reaction. Then oxidation of aldehydes and ketones. See, glucose is an aldehyde molecule, fructose is an ketone molecule. So those molecules are metabolized, are biotransformed through which processes? Phase 1. Phase 1 madli kutli process? Oxidation process. Now, हे झालं oxidative type of phase 1 metabolism. Now, reductive type of phase 1 metabolism involves aldehydes and ketones, nitro and azo compounds, and miscellaneous reactive reactions. Then, a third type of mechanism which is going to involve in the phase 1 metabolism is hydrolytic reactions. And hydrolytic reactions are hydrolysis of esters and amides, hydration of epoxides and arene oxides by epoxide hydrates. So these type of reactions are done or are going to give by the phase 1 processes. Now, type of processes or type of reactions given by phase 2 mechanism. Phase 2 mechanism generally involves synthetic and conjugated reaction. Conjugation means formation of new bonds with the new moieties. So what type of conjugation? Glucuronidation. Glucuronic acid बरोबर conjugate bond तयार करण्याची process, that is glucuronidation. Sulfate conjugation. Sulfate enzymes are involved. Conjugation with glycine, glutamine and other amino acids. So a drug molecule going to combine with amino acid present in our body. What type of amino acids? Glycine, glutamine and other amino acid. Then a conjugation, that is glutathione and mercapturic acid conjugation, formation of acetylation bond and methylation. So these processes or these type of reactions are involved in phase 2 biotransformation procedure. Now we are going to discuss the individual type of oxidation, that is phase 1 oxidation. आता phase 1 oxidation मध्ये काय काय कुठल्या कुठल्या प्रकारचे चेंजेस होतात? So those are oxidation of aromatic moieties. This type of process involves oxidation of aromatic drug molecules. This type is a major route of metabolism for many drugs, because many drugs are going to have this type of structure, this type of arene rings in their molecule. Many foreign compounds containing aromatic rings are susceptible for aromatic substitution, because large number of drug molecules which are available are going to have the presence of aromatic ring in their structure, आणि त्या सगळ्या drug molecules metabolism oxidation of aromatic moieties ने होतं. Now what type of reaction they are going to go for? First of all, it is hydroxylation reaction. So the oxidation of aromatic compound, that is arene, to their corresponding phenolic metabolite. When the drug molecule is undergo first of all aromatic oxidation, so it is converted into its phenolic metabolite, and that phenolic metabolite is known as arenol through an epoxide intermediate. म्हणजे large amount of drug molecules have this type of structure in their ring, and these metabolites convert into a phenolic metabolite by which process? Oxidation of aromatic moieties. आणि या प्रोसेस मध्ये conversion of arene to arenol हे होत असताना there is formation of one intermediate product, that is arene epoxide. Now this type of oxidation of aromatic moiety is known as hydroxylation, and this type of reaction is generally proceed at mid position, para position. See, this is very important one. They may ask in the GPAT example, so they may give the such type of molecule in front of you, and they may give the options that what type of metabolic reaction it is undergo. So what type of reaction? Oxidation of aromatic reaction converted into its phenol derivative, that is arenol, through an intermediate step, that is arene oxide. So this is first aromatic moiety oxidation. Now, for example, we have taken a drug molecule, that is containing an aromatic ring in its structure. So they are going to convert, or they are going to proceed by same aromatic hydroxylation mechanism at para position. So such type of drug is available in the market, that is amphetamine. See, amphetamine is going to have a same arene type of ring in its structure. So it is going to metabolized through aromatic moiety, oxidation of aromatic moiety type of metabolism, and converted into its phenol derivative. So such type of metabolic reactions are given by this. Then the next drug molecule is phenylbutazone, going to have the aromatic ring in its structure or benzylic ring in its structure. It is metabolized through its hydroxylation. Then metabolism of phenytoin, conversion of into its hydroxylated derivative. So such type of metabolism reactions are there for the first, that is aromatic hydroxylation reaction. We will see the remaining